In today's program, we're going to specifically compare mitosis and meiosis. Uh, we'll go over the differences and again talk about meiosis and genetic variability, and so why it does that, but also what cells are doing this and how it produces viability and how it can go wrong. So I have a Venn diagram there. That's a really nice aid for comparing mitosis and meiosis. So in the circle on the left there, the yellow, you have features of mitosis. In the middle, the kind of lens-shaped green area, you have the similarities. And on the right, in the blue, you have the differences. And so both mitosis and meiosis, we go through, we start with a diploid cell that has replicated DNA. And we're going to go through the same basic steps: prophase, metaphase, anaphase, and telophase. And we're producing new cells, but in mitosis, those cells will be identical genetically. They will still be diploid, have 46 chromosomes, and in mitosis, it's going to be for growth and repair of tissues. This happens in a wide variety of body tissues. Virtually all cells undergo mitosis. Meiosis, on the other hand, is a conversion of diploid cells to haploid. And instead of one round of cell division, there are two with no DNA duplication between them. Okay, and the resulting cells have 23 chromosomes. They're haploid, and they are all genetically distinct. And so this happens because of the pairing up of the homologous chromosomes and the exchange of segments, and then the random alignment of chromosomes in metaphase 1. Meiosis is very restricted. Okay, it doesn't happen in the vast majority of body cells, only in germline cells that are going to produce sperm and egg. So only in the ovary and the testes, and even then, not every cell in the ovary and the testes. So this is part of this life cycle where a fertilized egg that's diploid will go through mitosis, and they'll go through development and growth to produce us, an adult body form. And all the cells in our body will have 46 chromosomes unless you look at the cells in the testes and the ovaries. Some of those cells will undergo meiosis. Okay, and so they'll go through that 2 rounds of cell division and produce haploid gametes, the sperm and the egg. And so this is a distinct part of our life cycle. When we compare them directly, what happens is pretty clear in mitosis. Duplicated chromosomes line up on the metaphase plate, okay, and the microtubules attached to from either pole attached to either side of that chromosome, and then the centromere comes apart, and half of that duplicated chromosome goes one way and half goes the other. And so that ensures that each new cell will get one half of the replicated chromosome. And so the cells wind up with a diploid number. They start with a diploid number of cell chromosomes, but they're duplicated. And then they wind up with a diploid number of cells not duplicated. Okay. In meiosis, much of the differences between meiosis and mitosis occur in meiosis 1. In meiosis 1, in prophase, instead of chromosomes just condensing, chromosomes condense and they find a homologous chromosome. So they pair up with a duplicated homologous chromosome. So instead of seeing single duplicated chromosomes that look like an X, what you see are two duplicated chromosomes. So it looks like two X's paired together. And this is when crossing over occurs, and so the chromosomes exchange segments, and so they become genetically mixed. And then in metaphase 1, at the metaphase plate, instead of having single chromosomes line up, you have duplicated uh, pairs of homologous chromosomes line up. And so those pairs are going to be pulled apart. The centromeres will not split in meiosis 1, but the homologous chromosomes will. So at the end of meiosis 1, the cells wind up with duplicated chromosomes still present, but only half the number. Okay, so they've become haploid, but now they have haploid chromosomes that are duplicated. And so there's no DNA replication between meiosis 1 and meiosis 2. And in meiosis 2, it's very much like mitosis in that those duplicated chromosomes line up and are pulled apart, and the resulting sister chromatids separate. Okay, and so you wind up with haploid cells with non-duplicated chromosomes at the end of meiosis 2. Okay, so in meiosis 1, one of the big things then is crossing over, where the homologous chromosomes pair up, they line up next to each other, and they exchange segments. Okay, and in the other big thing that happens in meiosis 1 is that the homologs separate. And so those homologous chromosomes that have changed segments are what line up in metaphase 1, and they line up on the metaphase plate. So you see those two X's lining up, and the homologous chromosomes will be separated. And how they line up is random. And so there may be two ones that are mostly from the maternal line, or two ones that are mostly red on one side, or two ones that are mostly blue, or one of each. It's kind of random. And so you can have kind of a random separation of them. And then so this also mixes the genes as well as the crossing over. And so all of this produces lots of variation. And the more chromosomes you have, the more different possibilities. And so if you have 2 to the n possibilities, and if you have 23 chromosomes, that's like 2 to the 23rd power, or 8,388,608 possible gamete combinations. And so this really means that there's a ton of genetic variation that's produced through crossing over in this independent and random assortment of chromosomes. So this is really where we get all this variation, and it's why brothers and sisters never look identical unless they're identical twins, because there's this mixing of the parental genes. Last time I mentioned non-disjunctions, and so when I talked about non-disjunctions that produce Down syndrome, okay, but other non-disjunctions produce less distinct but still important conditions that are abnormalities that occur when when chromosomes don't separate. And I really didn't show you kind of what happens there. And you can have non-disjunctions in meiosis 1 or meiosis 2. Sometimes the homologs will fail to separate, and so you'll get 2 homologous chromosomes going to one side and none going to the other. And so you see that on the left here, that's going to produce an unusual number of chromosomes in both resulting daughter cells. So you'll have either an additional chromosome in some of the gametes, or missing one chromosome in a number of gametes. And again, usually this doesn't lead to a viable embryo, but it might if it's chromosome 21 or if it's one of the sex chromosomes. But you can also have non-disjunctions in meiosis 2. In meiosis 2, it would be that the centromere doesn't split, and so a whole duplicated chromosome goes one way. I mentioned these produce, if they're the sex chromosomes, these produce individuals that have certain syndromes where they're basically intersex, and these are fairly common, about as common as Down syndrome, but they're less easy to recognize. But they're common enough that we should think about 1 in 1,000 people may have this. And so there's, 25 intersex individuals in Freeport because we have 25,000 people. And so we would expect about 1 in 1,000 births. Um, and in large cities, this would produce large numbers of people who are intersex, not fully fertile males or fully fertile females. And there are other ways of producing this. And so sometimes you'll hear a politician or a religious figure say, you're either a man or a woman. There's nothing in between. And that's not how biology works. Biology has exceptions to everything, and things are sensitive to development. And so there are intersex people, and not only for non-disjunctions, there are other reasons as well. Okay, next time we're going to talk about patterns of inheritance, talk about the mystery of how genes are passed on and how traits pass from one parent to the next generation, and we'll go from there. Okay, see you next week.